in the past, and that is that it's now generally considered that microbes that live on and within us in many cases have a mutualistic or beneficial role for us. And this is a classical ecological relationship that exists throughout nature, where two different species, and in many cases completely different kingdoms like animals and microbes, depend on each other for health. And as a consequence of the explosion in interest in microbiome over the last decade, there have been many claims regarding effects of the microbiome on health status. And these claims come from observational studies based on sequencing and computational resources, which are trying to attribute certain communities of microbes with certain health conditions, such as neurological disorders and cardiovascular disorders and so forth. And these are very intriguing, has received a lot of news because of the excitement about being able to intervene in health conditions through maybe more amenable stem cell therapy, if you will, if you consider the microbiome a stem cell. But as is true for all observational studies, you're all aware that there can be confounding variables that bring this direct association into question. And that is probably more true for microbiome observational studies than almost anything else. True causation between health status may be really affecting many individual behaviors, diet, use of antibiotics, etc. And then those behaviors influence the microbiome. So the association between the microbiome and health status is actually a spurious causation. And what we're looking at are indirect associations. So because of that, we run the risk of wasting a lot of resources and not paying attention to a lot of critical information. So as dermatologists, I have no need to remind you, of course, that the ecological relationship between ourselves and microbes is not only beneficial, there are many hostile ecological relationships. So getting the immunological association between a microbe and us correct is really, really very, very, very critical before you start making interventions. And many of you may know the fecal transplant story, which really started with exactly just the observational study saying, oh, this is the bugs in healthy people poop, and these are the bugs in C. difficile people's poop. And if we just move randomly the bugs over, they'll do better. And there was some early success, quite a bit of success, but there was also some fatalities as a consequence of moving organisms somewhat blindly from a healthy individual that has potential for causing pathology without really knowing what's going on. So the way we've approached this problem is somewhat differently than the observational studies, and that is to really try to understand the molecular interactions. What is the specific molecule that a bug could be releasing that might have benefit based on, say, the specific receptor in the human ecology. And then once we have those, which are still just hypotheses, begin to start to conduct control, randomized, blinded studies to determine whether or not the microbes can be really used as an intervention therapeutically for human skin diseases. So most of the microbiome literature that's out there is related to fecal microbiome, the gut. And if you look at PubMed, there are over 6,000 publications on gut or fecal microbiome, and relatively fewer, only about 500 or so, on the skin microbiome. So why is that? Well, part of it is funding, but a large part of it also is ease to do the studies. There's a lot of bugs in poop. So it's pretty easy to do a sequencing reaction on stool and specifically amplify or directly sequence bacteria from stool. Whereas in skin samples, despite how accessible it is for swabbing, when you do a swab, most of the DNA that you obtain from the skin surface is human DNA. So trying to pick out the relative microbial DNA is much more difficult. And of that, it's easiest for bacterial and eukaryotic DNA associated with fungi, and most difficult for viral, although all three components, bacteria, fungi, and viral, are all part of our microbiome. And then there's the conceptual idea of how a microbiome might interact with these two organs. The intestine is primarily thought of course, as an absorptive organ. So what's in there, we're trying to selectively extract the goodness from, if you will. Whereas the skin to the general population is often thought of as a barrier organ, of course, and if the barrier properties don't operate effectively, we're in trouble. But if you look closer as a cell biologist or an immunologist at the intestinal interaction with microbes, you realize that these associations are very different. The intestines does everything it can to try to separate itself from the microbes themselves. So the villous surface of the intestinal epithelia has actually very little contact with microbes. There's a very thick antimicrobial rich mucus layer that covers this. So this large surface area that you think about for absorption of metabolites from the lumen of the intestine is good for metabolites, but it's not in contact with the microbes. The microbes are separated by this mucosal layer. So the interaction, we think, most of the interactions that are beneficial between the intestinal and gut microbiome have to do with metabolites that might be then absorbed. And that's very different than in the skin. And a few years ago, while thinking about this, we realized that the published surface areas of the skin acted like the skin was a flat sheet of paper and had no invaginations. It was basically just ignored. And if you went and calculated based on about 1 millimeter or so depth and about 20 microns diameter of each hair follicle, you could realize very quickly that our skin surface area is at least as large as the intestine, and if you take into the mucosa into account, it's much larger. So we are a massive barrier with a massive absorptive capacity if we want to. And even more so, if you look in hair follicles, under normal conditions, these follicles have about 100 times more bacteria within each of these large surface areas than on the surface in the interfollicular distance. So they're abundant in there and they're highly protected. And then finally, bacteria on the skin don't just sit there on the surface. They have proteases, and there's a penetration through the epidermis into the dermis. It's a small fraction, but about 1/100th to 1/1000th of the bacteria that exist on the surface of the skin are normally in contact with our circulation and deep in the dermis. Now, if you think about it, the starting concentration of bacteria, about 100,000 bacteria per centimeter squared, that means you're getting about 1,000 bacteria in every centimeter that are interacting with us directly. Then think about the genome and how to figure out how to understand what's going on. Well, the human microbiome has clearly many species and strains that are associated with it. And if you count up all the genes there are over 1 million different genes. Many of the elements of the skin microbiome are transient, may have nothing to do with us. And that contrasts that to the human genome, which has a very small number of genes. How to figure out what interacts with which, what's spurious, what's reality, what's potentially pathogenic is not only like trying to find a needle in a haystack, but it's like trying to find a needle in a needle stack. It's a difficult problem. So I'm sorry. So the way we've tried to unravel this is to do this simultaneously in 2 different ways. First, use classical genomic approaches where we're sequencing both the DNA as well as the RNA from the microbes so we understand who they are and what they're making. But at the same time, also try to recover as many as possible that are culturable, which in some genuses is pretty easy, others more difficult. And then culture those bacteria and do mass spec analysis and functional analysis, try to actually look and hypothesis test things that the bacteria are making and how they may benefit human skin ecology. And when you make those hypotheses, you can then go back and look at the metagenomic data, try to correlate that, and gain a greater understanding of what is helpful and relevant to human skin diseases. So can we answer the question of how microbes influence skin health? And we started off in a very simple direction, primarily really an immunologist, and we began by thinking about how a keratinocyte or epithelial cell interacts with the pathogen. And then when you can think about how the pathogen and maybe the commensal interact with each other and how the commensal interacts with the skin. So simple two-way questions you can answer. Our very first question was to look at how the skin can protect itself. And that's where my laboratory began understanding the innate immune defenses for hosts and pathogens. And we continue to be very active in that regard, have discovered things that have become known as antimicrobial peptides or cathelicidins. And essentially what these are natural antibiotics. It was actually somewhat surprising that this wasn't better appreciated at the time when I was a postdoc, because it seems now in retrospect so obvious that we would make natural defenses against bacteria. We proved it by knocking it out in a mouse, and when a mouse no longer has just one particular antimicrobial gene known as the cathelicidin antimicrobial peptide, or CAMP, it then became susceptible to the group-A strep infections. We continue to work on this. There's a lot of other associations that are interesting, and one of the most exciting has been realizing that these human genes, these human AMPs, are not only just acting on bacteria, but like many human genes, they're tasked to do multiple things. We only have 20,000 or so genes, and we have a lot of biological processes. So it's typical for one gene to do more than one thing. And that's very true for the host antimicrobials, where we found several years ago that these same things at normal levels, they can protect against pathogenesis. If they're turned on inappropriately and too high, they also drive an immunological response, an inflammatory response. And in particular, the human cathelicidin LL37 drives a vascular inflammatory response that seems to explain much of the pathogenesis of rosacea. And I'll just mention as an aside, some very exciting data that suggests that some of the functions of LL37 have actually been usurped by the spike proteins on the surface of SARS-CoV-2 virus. And we've got our first set of experiments in the lab, despite the shutdown, suggesting that this hypothesis is true, and maybe this nasty virus is actually using some of the pathways that we found in rosacea to make inappropriate damage to the skin, well, to the lung, I should say. As a clinical pearl, I just want to point out that in rosacea, the older classification system of trying to separate erythematous from papular, from phimomatous and papulopustular is been abandoned. We've now moved to a new classification system based on phenotypes, recognizing that individuals can have more than one of these phenotypes, and this will help us in future classifications and assessments of therapy in rosacea. So I just remind everybody that the PPP and ETR forms of rosacea have now been abandoned, even though they'll still be in the textbooks. So what are other things that the antimicrobials can do? Peter Elias, some years ago, with a medical student from UCSD, Karen Aberg, went on to do a residency up at UCSF, also found that the host antimicrobials act on keratinocytes to help them form the barrier. And what he found is in the knockout mouse, with this supposed antimicrobial, which I just told you can also cause too much inflammation, it also had barrier defect properties. So this, as you can see, becomes a very complicated system with a lot of biological processes that we need to understand before we start intervening in the expression of these things. So what about another simple interaction? We looked at how the host can affect microbes and discovered interesting things. What about how the microbe can affect hosts? And a graduate student of mine, now about 20 years ago, Anna Kogan, who's now completed her dermatology residency at UCSF, helped us ask the question, is it possible that bacteria are beneficial for us? And at the time, a decade ago, this was a bit of a crazy question. And what she found was that it could. And then some work taken up by a postdoc of mine at the time, UPY, found that one of the things that commensals do is they help prime the surface so that it can increase the antimicrobials and protect against infection. So this relationship, Yu Ping found specific molecules later on in her laboratory about how a commensal product can stimulate the skin to then make something that the skin uses to kill bacteria. And this is a promising line of investigation that she continues. What about how the host can affect the immune response? So that's the work that I was talking about from Anna Kogan and Yu Ping Y, and led to really the first paper that showed any specific anti-inflammatory relationship between bacteria and humans. Was later reproduced in the colon. But again, we continue to do this and understand how it works, and who knows, this may even have benefit in the SARS-CoV-2 pandemic right now. So now I want to go, as this story unfolds for you, go to another simple relationship. We asked how host affects the pathogen, how the commensal affects the host. What about the commensal and the pathogen? And this is one of Anna Kogan's experiments that very simply illustrates that the bacteria on our skin can also directly kill the pathogen. And what she did here, these are her fingertips. She washed her hand and coated it with a low virulent strain of group A streptococcus. And group A streptococcus, as many of you may know, is beta hemolytic. So if it grows on a blood agar plate, it'll cause the blood, the hemoglobin, to lyse. So you can simply see the group A strep by the lytic action on an agar plate. Then she did the same thing, washed her fingers, but now just repopulated with staph epidermidis. And you can see the staph epidermidis is there because it makes all these little dots, but it's not hemolytic. Well, what if your skin now wasn't cleaned of most of the surface commensals, but was highly populated with staph epidermidis as here, but then exposed to the same dose of group A strep as she exposed herself in this experiment? And this is the result. So you see that the presence of the staph epidermidis is inhibiting the ability of group A strep to survive on the surface of the skin. So this leads to interesting questions, right? Especially now in a day of pathogen transmission. Is it possible that we're washing our hands too much and removing natural antimicrobial barriers, that if those helpful microbes were there, could they be protecting against bacteria and viruses? We actually really don't know the answer to that question, but happily, I'll tell you that when you wash your hands, even with a surgical scrub, very thoroughly, if you go back 10 minutes later, the bacteria are back to normal, even if you don't touch anything. And we think that's because of all those follicles protecting your microbiome, and they're very quickly able to reemerge. So some interesting questions about hygiene. And then in collaboration with Tisa Hada's clinical trials group, we're doing some similar work now with Rady. Teru Nakasaki took this a step further and started looking at just how frequently the bacteria on our skin can fight against a pathogen. This is a plot. Each dot is an individual, and he's looking at whether the fraction of bacteria on the skin can kill Staph aureus. And on normal individuals, non-atopics, most of us, almost 100% of our bacteria can. But interestingly, the atopic, even non-lesional skin that was healthy appearing is colonized by bacteria that cannot kill Staph aureus. If you plot that based on the amount of Staph aureus that you detect on people, you see a remarkable clinical association, which you very, very rarely see. So this is about 100 patients. It's not a giant study, but it's not a tiny study either. And those individuals that had over 50% of the antimicrobial coagulase negative staph, that's what CONS is, if you have enough of those, they never were colonized by Staph aureus, just zero. Whereas, of course, those with a low amount, many of them were colonized by Staph aureus. And even in the non-atopic healthy population, we incidentally picked up a few people that were colonized by Staph aureus, even though we didn't know it. And of those 3 people in this study, all of them were absent these antimicrobial CONS. So a very exciting observation, just I think in a way proving how important those topical bacteria can be. And this is a recent time-lapse video that Teru made in the lab. This is an agar dish. It's impregnated with Staph aureus. It's going to start growing when I start the movie. This is a single colony of a CONS bacteria, a Staph epidermidis, that is from an atopic patient. It's the same species as, what we think to be a healthy species as this species from a healthy individual. But there's a very important difference, which you'll see when I run the movie. So this is a 24-hour time lapse. The Staph aureus is growing up, and you can see, I think, quite dramatically that the bacterial colony that's harvested from a healthy individual has actually secreted something that is a potent inhibitor of the ability of Staph aureus to grow. It was fun to make little movies. That's why we made this. So let's see if I can advance it, however. Okay. So in the first part of my talk, what I've tried to do is answer the question, how does the skin protect itself? Well, we know about antimicrobial peptides that kill bacteria. These antimicrobial peptides can activate the host immune response. Sometimes that's good, but sometimes that's associated with disease. Antimicrobial peptides can enhance our physical barrier. The commensal bacteria can induce the AMPs. The commensal bacteria can alter the host immune response. And now the commensal bacteria kill other bacteria. So we've got to think about human skin health by paying attention to many of these other interactions that are ongoing. But to really understand the system as a whole, what you need to do is not only these very simple one-directional interactions, but think about the complicated system as it exists as a whole. Because you have many more than 2 types of bacteria, and you have many more than just keratinocytes interacting for host cells. And each of these we know will interplay in a different way. So over the last 4 or 5 years, we've tried to tackle this problem, a much more complex, multifactorial problem. And it's a big challenge. So the way we've done that is, instead of having a monodirectional interaction, let's start doing bidirectional interactions, because like an antibody, that allows you to expand the bidirectional interaction into a complex system. So our question is, are microbe interactions and host microbe interactions resulting in change overall in the system? And the first part of that, what about an aspect of the pathogen-host interaction that we can understand? I mentioned earlier that Teru had shown some years ago that microbes on the surface, which are illustrated here in green, have the capacity to burrow through the epidermis. Here, the nuclei are stained with DAPI, and the microbes can enter. If the microbes are dead, they can't do that. So this is requiring a live metabolic process, and it's due to proteases that the bacteria produce. So Mike Williams, who's now a postdoc in my laboratory, but at the time was a starting graduate student, asked the question, is the microbes doing more than just producing their own proteases? Are they activating the keratinocyte proteases? And the answer to that question, I wouldn't be bringing it up if it wasn't true. This was the subject of his PhD thesis, was yes. And actually the Staph aureus, which penetrates particularly well as products, can induce a number of kallikreins in the skin. These are serine proteases, things like kallikrein 5, kallikrein 7. these serine proteases are dramatically induced by something produced by Staph aureus. And he went on to define exactly what that was. And these are these small molecules known as phenol-soluble modulins, PSMs. There are a number of those, and Mike was able to define specifically which PSMs are most active to activate keratinocytes. And then he was able to look at the genetic response of the keratinocytes by transcriptional analysis and sees a whole bunch of epithelial homeostasis genes are being basically twisted by Staph aureus. Staph aureus makes some of this PSM and makes our keratinocytes essentially go crazy. So it's not just an infection here, just the colonization without infection leads the skin surface to be highly proteolytic because of the presence of these PSMs. You can see the PSM right on the surface of the atopic dermatitis skin, and you can see the trypsin activity on the surface of patients with the skin directly correlates with the amount of the PSM that we see there. So some very nice clinical observational correlations. I'll highlight this one little bit because I'm going to come back to this at the end of the lecture. There are a few situations where we also notice other bacteria present there, like Staph hominis. And Staph hominis seem to be associated with a very low capacity of Staph aureus to make the PSM. This is a further illustration. Those PSMs are important now using mice where we can intentionally expose them. This is a mouse with no bacteria that's just been shaved. This is a mouse that we put Staph aureus on a gauze pad. You can see it highly disrupts the epidermis. Here's the same amount of Staph aureus, but Mike was able to genetically delete just that one gene, and you see that in the absence of that gene, it couldn't damage the skin, even though the Staph aureus was there. And here, the Staph aureus has the PSM, but doesn't have its proteases. So it needs both. It needs the proteases and the PSM to do its damage. And it makes a very highly inflammatory response, but in some cases resembles some human skin diseases, which I'll get to in a moment. So the first interaction is we see Staph aureus is producing molecules that's changing the genetic behavior of our skin surface. But what about if it's doing that now in the presence of other bacteria? How do other microbes influence the ability of Staph aureus to influence us? And this turned out to be a very exciting observation, and we were helped greatly by our collaborator at University of Colorado, Alex Horswell, and it's exploiting something known as quorum sensing. Quorum sensing is what goes on when bacteria come to a high density. And many pathogens like Staph aureus, when they get to a certain density, they change their genetic composition, their expression, so that they start doing something different. They make biofilms when the quorum sensing is activated. And one of the things we knew when we read about these PSMs and these proteases was that when quorums are reached for Staph aureus, they start making these things that are damaging human skin. So Mike wanted to know, by this strategy, is there something else present in the microbiome that could negatively affect the capacity of Staph aureus to use its quorum sensing system? And by screening different isolates from the surface of human skin, we found that some, but not all strains of the common coagulase negative bacteria, things like Epi and Hominis, Warneri, these are things we all have on us. They made something that inhibited the activity of the quorum sensing gene AGR. Once we knew the strain, we could genetically approach them and look what's different about them and define specific genes and what they encoded, these molecules known as auto-inducing peptides. And when we found these novel auto-inducing peptides, we could synthesize it and then use the synthetic auto-inducing peptide made by, in this case, it was the Staph hominis, to show we could turn off Staph aureus. Now you could do this, the Staph aureus grows. There's no selection pressure here against like an antibiotic. If you treat an organism with an antibiotic, there's an ecological pressure to evolve resistance. Here, the Staph aureus is fine. They're surviving fine, but they can't make the toxins that damage us because their quorum sensing is turned off. And this is an illustration of this. This is an atopic dermatitis, a Staph aureus clone. It's making PSM. We see that in the Western blot. Now you add the Staph hominis, the same amount of AD38, and the PSM is gone. Here's what happens with the Staph aureus on the skin. This is another kind of assay where we can live look at the expression of Staph aureus by the presence of a bioluminescent tracer. If the hominis is there, the Staph aureus can't do it. And here is that same kind of assay as we showed before. Now we're not genetically modifying the Staph aureus in any way. We're just putting the Staph hominis here. This Staph hominis can't kill Staph aureus. So there's just as much Staph aureus here, as we can see, but the ability of the aureus to damage the skin is dramatically decreased. Transepidermal water loss is decreased. So question number 2, we're making some progress on in the multivariate analysis. Can we unassist them? Yeah, we're starting to, because we know there's some bacteria that are making things that when they coexist with maybe a bad guy, they can inhibit that bad guy by inhibiting toxin production. And as I showed you earlier, they can also, other bacteria could kill the bad guy. So there's many ways that the microbiome as a whole interacts to potentially protect us. What about the other way? What is the host doing to interact with the microbiome? So here's a disease that's familiar to all of you, Netherton syndrome. And I kind of have dropped a little hints along the way when I mentioned things like callicrins and serine proteases. And as you all know, and I hope all the first-year residents know that now as well, the mutation in Netherton syndrome is for a gene called SPINK5, and that encodes LECD1. And what is LECD1 but a serine protease inhibitor? And the pediatric dermatology faculty understand very well that human serine proteases are very important in newborn skin for managing the epidermal barrier. And when infants are born with a mutation in SPINK5 and absent functional LECD1, there's a severe neonatal phenotype. So clearly this serine protease inhibitor is important for interacting with the host. But as our patients with Netherton syndrome grow up, we observe that there's lots of areas of the skin that actually seem to eventually differentiate normally and have intact barriers despite the absence of the enzyme. And there's patchy regions where the barrier is broken down, and that barrier breakdown can be transient. We can see flares coming on, going, coming on, going. Well, what we've learned from Mike Williams and Alex Horswell's observations for microbiome interactions for proteases, we wondered if this interaction may play into it as a whole. So we conducted a study with Alain Hovanian in France. He's one of the world's leaders in Netherton syndrome. He's the guy who actually cloned the mutation and led to us understanding about how SPINK5 causes this. And we looked at many individuals for Netherton study anyway. We had 10 NS patients over time, and different individuals we had different number of time points for how severe their disease was and what their microbiome was like. And it was shocking to us that every single patient that we looked at was heavily colonized with Staph aureus. And this was not something that was generally understood in the literature at the time. You think about Staph aureus colonization in atopics, maybe 25, 50% of the patients have Staph aureus in AD, but in Netherton's, 100% of them have it at some time. If you look at the amount of Staph aureus, you can see in a healthy individual, typically we won't see any Staph aureus on a swab, but from even the non-lesional Netherton subjects, and even more so the lesional, we had a lot of Staph aureus present on those individuals. Then in collaboration with Chris DuPont at the Craig Ventnor Institute here, with that much microbial load, we're able to do something called shotgun sequencing, where we sequence every single gene from the bacteria, not just to identify their species, but to understand what genes they had. This is an illustration of how complete the genomes were of these bacteria. He told us at the time, and this is pretty recent publication, this is the most complete genome sequencing of the human skin microbiome that was done at that point. And he tells us that based on the genomic composition of Netherton's patients, their microbiome is very, very distinct. They are more different than healthy skin microbiome than the atopic microbiome is from humans, and much more different than psoriasis is. And what he picked out in there was that in addition to the Staph aureus, there are a few really weird Staph epidermidis strains. We wondered what that meant. So Staph aureus, big part of the genome, but Staph epidermidis, the second most abundant. And the Netherton's patients, if we looked at their lesional skin, their genome, a big component of that, not found in the healthy skin. And when we looked at those toxins that Mike described, we found that the Netherton's patients in the non-lesional and the lesional skin had a lot of those protease-inducing peptides. And that correlated, the amount of peptide we found on Netherton's correlated exactly with the amount of Staph aureus, and the trypsin activity on the human skin correlated with the amount of the toxin that was present there. And then this was an experiment that was done by someone who's going to be one of our residents next year, Shadi Kahil, when he rotated through my lab last year. We wondered, if you took a normal human keratinocyte and made it into like a Netherton's-like keratinocyte, knocked down SPINK5, what would happen? Well, under control conditions, as expected, in the absence of SPINK5, there was more trypsin activity. But then when you added this abnormal microbiome component, it greatly elevated that. So our hypothesis is that under this condition, you now break a threshold where the trypsin activity gets so high, you don't have the host protease inhibitor to slow things down. Now the barrier begins breaking down. So what about this one patient here, some of you may have noticed, that didn't have as much Staph aureus? Well, this was the patient that had a lot of Staph epidermidis. And it led to another postdoc in my lab, Laura Cow, to start a project looking at Staph epidermidis proteases. And epidermidis, which we usually think is a good guy, turns out sometimes can be bad, can make too much protease. And again, using these same kind of genomic techniques in our Netherton's cohort, she sees that there's one patient that was an exception to the Staph aureus rule on one visit, was very, very high Staph epidermidis patient. And using the same kind of logic, trying to understand the molecule, we figured out that it was this protease called ECPA. We deleted that one gene from Staph epidermidis. That decreased its protease activity. And doing the same kind of experiments on mice that we did before with Mike's work in Staph aureus, a Staph epidermidis strain that makes this protease gives them a rash. If it is present on the skin without ECPA, it does not cause the inflammation. It's a dose-dependent effect. So the more epidermidis you place on the skin, the worse it is. The more epidermidis you place on the skin, the more it causes transepidermal water loss. And if you kind of look at these rashes that this particular protease makes, I wondered if she was actually recapitulating ichthyosis linearis circumflexa that you see as a characteristic of the Netherton's patient. So is it what we're looking at here? It's a human skin disease. It's not an infection, but it's a skin reaction to an abnormal microbiome. So that may be true. And it may also be true for atopic dermatitis. So this is another observational study we've conducted. We just sent the paper back in. Hopefully we'll get it accepted soon, looking at the atopic population. Those of you who think about atopic dermatitis, and I know most of you do quite a bit since it's such a common disease, realize that Staph aureus is only maybe a minor subset of many of your atopics, but they often have a lot of disease. And if you culture them, the lab tells you no pathogens. They won't tell you that they're loaded with Staph epidermidis. And that seems to be what's happening, actually. This is a log scale of the amount of Staph epidermidis that recovered from a swab. And you see that AD lesional skin has 10, 100, 1,000 times more Staph epidermidis than healthy skin. This correlates very carefully with the EASY score from patients. And the amount of just that gene I was talking about, that protease, also has a very significant correlation to EASY. And so putting it all together, another piece of this puzzle here, the host interaction with this, at least one of them is something we knew before the protease inhibitor LECT1. We used to think about it only LECT1 across the keratinocytes, which is still true, of course, but we also think this protease inhibitor is interacting with the microbiome. So for Netherton syndrome, we have a novel pathophysiology, and that is that the Netherton's patient is colonized with an abnormal microbiome. I should update this, Staph aureus and Staph epidermidis. That makes more of the kallikreins. When they make more kallikrein, the absence of the LECT1 to inhibit the protease leads to barrier disruption. So we think this is an important part of the interaction and suggests that maybe interventions into the microbiome of Netherton's should be something that's tried more frequently when caring for these patients. So last question, and I think most important is, with all this stuff that we've learned, and this is really 20 years worth of work, I've tried to highlight some of the people in the laboratory who've made key contributions, but there have been many more. Can we do something to intervene with this? And classical approaches to diseases like atopic dermatitis have been barrier repair and immunosuppressants, either topical or systemic immunosuppressants. And if you think about this as a system as a whole, that's a good thing to do because that's part of the disease. But what if the microbiome was really a significant part of this? Is there something we could do, like introduce a healthier part of the microbiome to improve this disease? Could that introduction of a healthy new element cause a shift in the microbiome that not only transiently improves disease, but permanently cures these patients? So our model in AD, from what I've told you thus far, is that there are two kinds of bacteria that, at least, that are doing things that damage the barrier. Staph aureus is making proteases and PSMs. Staph epidermidis is making ECP. That leads to changes in the barrier, which is bad for derm. We know that there are some Con-S antimicrobials that produce things called auto-inducing peptides. And what's beautiful about these AIPs is that some of them act against many species, and they will inhibit this toxin production. Also, because they're not antimicrobial, they're not subject to selection because of their action as an antibiotic. And then there are also Con-S, as I illustrated in that movie, that make antibiotics that could help us as well, although we worry about antibiotic resistance developing and how that can happen. And then if you got just the right bug, maybe you can find a bug that does these things that act against the things we don't want, but also permits the rest of our normal microbiome that we're only still trying to understand to survive. It doesn't have a negative effect on the beneficial organisms like all of the pharmaceutical antibiotics would have. So when you treat a subject if you're able to get an effective treat with mupirocin or an oral anti-staph antibiotic, you not only kill this, but you kill this and you kill all of these. So when it begins to recover, the Staph aureus has nobody to resist it. We don't want that. We'd like to be more selective. And we screened a giant library of organisms for that and identified candidate bacteria that can do many of these things. One of them is called Staph hominis A9. And I'll just show you the mouse experiment here where we take an atopic dermatitis model mouse and apply Staph aureus and apply Staph hominis A9 and show that the hominis A9 is able to very effectively kill the Staph aureus. It inhibits that and it inhibits inflammation. Even more impressively, if you look at the Th2 cytokines induced by the mouse, you see that the Staph hominis co-application is inhibiting IL-4, IL-13, and something else called TSLP, which is a good marker of keratinocyte inflammation, really, really potently. And this is working as well as dupilumab for blocking Th2 cytokines. This is work that's being led in on the animal side of things by our fellow faculty members, Terry Nakasuchi, who's a project scientist in my lab, and our technician, Anna Butcher, is going to be with us for a few weeks before she starts medical school, hopefully live and in person in July. So that initial observations, they published about 2 or 3 years ago that describes the capacity to do this, and the strategy behind it. It was published in Science Translational Medicine. And importantly, that paper also studied a clinical trial that Dr. Hada's clinical trials group organized that was looking at autologous application of bacteria. We took one atopics patient, tried to find a good bacteria, put it back on, and saw that it worked. We've since conducted the study, and this is the last piece of data I'll show you, and it's not yet published, we're just writing it. One of my things I get to do in seclusion right now is to take this idealized hominis strain, grow it up, and apply it. And this was a multi-center, double-blind, placebo-controlled study. A screening phase identified only Staph aureus positive atopic patients. And then, since it was basically a safety study, we just smeared the bacteria on them twice a day for just a week, make sure everything's okay, and then followed them up in person 96 hours afterwards, and then called them back 30 days later, make sure nothing happened by putting these bacteria on them. And happy to report that as a phase 1 trial, it was incredibly successful. There were no increase in AEs compared to the vehicle when we unblinded it. In fact, depending on what statistical tricks you play, there was actually perhaps less AEs in the treated group than in the vehicle group, suggesting they were having less flares of redness and so forth in this very brief period. But since hominis did all these good things, we couldn't help ourselves to also take some swabs on these patients and to make some measurements to see if, besides being safe, is it good in some other ways. And if you look at the Staph aureus colonization, it worked very, very well. On both lesional and non-lesional skin, we reached highly significant at day 4 in these populations in both San Diego and in Denver. It dropped the Staph aureus as we did in autologous treatment. So that works well. And I'll tell you, works much better than the published studies for topical pharmaceutical antibiotics, which over time, in most cases, do not show any drop in Staph aureus, despite the fact mupirocin kills Staph aureus quite well. The other exciting part of the study is that if you look at the subjects that had that best drop in Staph aureus, the ones that were most sensitive and able to respond quickly, and you looked at their local EASI on their skin, the local EASI dropped on treatment, continued to drop off treatment, and reached statistical significance at the last blinded assessment time that we had off treatment. So there's some suggestion now that this cream could be used as therapy, and full disclosure, conflict of interest, Tara and I are inventors of this. It's a patent, and there's a local company that I co-founded called Matricist that's trying to do this in a larger study. The NIH has funded Dr. Hata and I to continue in this study. We just were recently awarded another 7 years of funding to conduct clinical trials and further mechanistic trials to better understand how the microbiome can improve AD. So I'll leave you with this, and that it's never one thing, but I believe, hopefully I've convinced you that human health depends on the genetics and environment as well as the microbial communities, and that can affect health status. But you have to test causation. There's a lot of spurious claims, which frankly I think have damaged the reliability of much of what is said is about the microbiome in general, and we have to approach this much more cautiously and with a critical eye. So with that, just to keep it short, I'll just put up a slide of many of the key people that were part of the story I told you, and also remind you that the SID meeting is going to go on this year, and many of the clinical audience may not be attending the SID, but this time, because of the pandemic, it's going to be virtual, so you'll be able to see both some live talks and some of this work presented as a recorded video, so it should be fun. So with that, I will end, and if there's any questions, try to take them. Thank you.
Thank you, Rich. That was great. We have one question. What are your thoughts about potential function of the appendix as a safe reservoir?
Yeah. Yeah, it's an interesting, and you may be aware that hypothesis has been thrown out there, and although it didn't seem to do any digestion, maybe it was a microbiome's sake, and seems pretty reasonable to me, but I'm not aware of data that's proven that.
Okay. Rich, I have a question. So we know that when one has inflamed atopic dermatitis, the diversity goes down and the staph goes up, and we had this simplified story that then when the skin got better, whether from topical anti-inflammatory or a systemic agent, that the staph went down and the diversity went back up. But isn't it true that when you're really inflamed, it's not just Staph aureus that's up, the Staph epi also is increased in quantities? How do you explain that, and what's your sense of the sequence?
Yeah, so I think the data I just showed you, to my knowledge, is the first demonstration that you could specifically attack Staph aureus first, and then without an anti-inflammatory, and then see easy improve. So I think the sequence is neither and both. It's a cycle. The bacteria, which, as I showed you, both epi and aureus, can induce inflammation, and then an abnormal immune response and an abnormal barrier can set up a situation where the bacteria can grow unchecked. And one of the other things we know is, like when you use dupilumab or when you use with a topical anti-inflammatories, and the barrier begins to improve, Th2 cytokines go down. What happens then simultaneously is the host antimicrobials transiently go up, the barrier improves, and those knock down the bacteria. So, dupilumab is an anti-staph agent, and topical triamcinolone is an anti-staph agent, both anti-staph epi and anti-staph aureus, when we're talking about colonization, not penetrated infections. And what we're trying to show now is that arm, which has been therapeutically beneficial, may also be assisted, if not superseded, by a non-immunosuppressive approach where we just try to normalize the microbiome. I think eventually we'll need to do both. Maybe a way to, pulse treat a severe patient with one immunosuppressive therapy and then get them off of that and maintain remission with the other. And that's the trials we're doing now.
Great. Looks like there's another question. Any prebiotics to promote growth of staph hominid A9, since abnormal staph epidermis and follicles could recolonize in the same 10 minutes it does after washing?
Yeah, you listen so closely. Thank you for staying awake. And you're right to use the term prebiotic and not probiotic. There's a lot of confusion about that. Probiotic, I intentionally didn't use the term because there's so much baloney about that, but it's meant to imply the live organisms. Prebiotic is basically the food for them. Is there food that selectively can improve the growth of the beneficial organisms over the non-beneficial organisms? There's a lot of work on that. I think in some systems there is an opportunity for that. To me, probably the best example of the proven prebiotics, if you will, is what you're already doing for your patients when you're treating them with emollients and maybe anti-inflammatories, is you're improving the environment of the skin in a pre-growth way where you're selectively allowing the pathogens to die off and the commensals to survive. But I'm not aware specifically of anything in a disease state where you could say, I add this kind of growth media and that's going to not let Staph aureus grow, but it's going to allow like a hominis to grow.
Okay, great. Well, we're beyond the hour, so Rich, that was fabulous. There is a QR code up now which you can use to put in your comments and or to document attendance. I don't know if Paula takes pictures of the participant list, which is another way of doing it when we were up in the high 60s today. Thank the residents for great presentations, and Rich, that was fabulous. Everyone stay safe and healthy and have a good week.
Thanks, and I want to point out to the UCSD faculty that after everyone logs off, please stay on the line. We're going to have a presentation by Dr. Kane. And the way to use this QR code is just hold your iPhone camera to it and it'll immediately take you to a questionnaire to prove your attendance. So thank you everyone for your patience in listening to my lecture, and congratulations to residents. That was a great patient presentation preceding it, as always.
Has everyone been able to check in?
Paula, can you hear me?
Yeah, Chris, we hear you. Thank you very much for joining. We're just coming off a couple of other contiguous Zoom meetings, so if you just give us another minute or two, Paula can confirm when we're just limited to UCSD faculty. Then I'll just start. Thank you.
Oops.
Okay, Chris, I know your time is valuable. I think we're pretty